this is Michelle from Bioscience Education Center. Today we're going to look into a new topic called alternative splicing. So I don't know how well you guys know about alternative splicing or splicing in general, but make sure you guys know that alternative splicing and splicing, it's not another type of splicing. Alternative splicing is an application of splicing. I feel like I'm saying splicing a lot in one sentence. Anyway, so what I'm going to do right now is that I want to kind of revisit the idea of what splicing is first. And once that's cleared up, then we can move along to talk a little bit about alternative splicing and the benefit of it. Okay, it's actually a very, very important process that allows us to make a lot of different types of proteins in our body without storing a lot of genes for all of these proteins. Okay, so we can limit the number of genes while making the maximum number. I wouldn't say maximum, but making like the most out of what we have. Yeah, that's better. Anyway, so let's go ahead and start a little bit on what exactly is splicing first. So splicing, I always love to start with definition. Splicing. Oh, by the way, I forgot to mention, this is actually a higher level content in IB Biology, Unit 7.2. So in that unit, we kind of talked a little bit about higher level transcription process, so a little bit more on the specificity of the enzyme direction, RNA polymerase, in terms of transcription. So after that, we also discussed a little bit on post-transcriptional modification, which is a modification of the pre-mRNA to turn it into mature mRNA before translation. And so alternative splicing is actually an application of splicing, and splicing is a type of post-transcriptional modification, hence everything is just in Unit 7.2. So if you're doing standard level IB biology, you can just skip this video and good luck with revision with like other units. This is so awkward. All right, so let's continue then. So splicing is the process of the removal of introns, these non-coding regions, meaning regions that are not used to make protein, okay? Removal of introns from the pre-mRNA and combining the exons, the coding regions together to form a mature mRNA. Yes, okay? So this process is done by splicism, and it happens in the nucleus. Okay, it is a type of post-transcriptional modification. And what post-transcriptional modification do is that it converts a pre-mRNA to a mature mRNA so that we can transport it out, so that we can transport this mature mRNA out for translation for splicing. So imagine this is the mRNA that we made after, the pre-mRNA that we made after transcription. Okay, that's the pre-mRNA. Within the pre-mRNA, there will be regions that are actually used for protein synthesis, and there are also regions. So these like green bits I'm shading here are the coding region, we call exons. However, within the pre-mRNA, there's also some regions here that I'm shading it in red, are non-coding introns. And the last thing that we want is to have some non-coding region within a coding region that, you know, ultimately it's going to affect the coding sequence on the mRNA, which means that you probably won't be able to make the protein that we want. So that's why it is very important to remove these introns and just remove these introns out to make sure the mRNA coding sequence is correct, it's accurate, that the ribosome will be able to, enabling the ribosomes to pick like the correct amino acid and put them together to make the desired protein at the end, all right? So how do we do it then? So I don't know if you guys heard about this, but the process is kind of like a little bit disgusting, but it's like kind of like popping a pimple. So it is done by something called splicism. So the splicism would be a binding somewhere the beginning at the end of the intron. Spliciosome. Okay, so they would come together and like popping the introns out like this. So let me draw this again. This is something that I tell my students all the time too, like popping a pimple. Okay, axon. This is the shaded axon. And then the introns will be popped out like this. Sorry about the scale. I don't know why it became a little bit larger. Anyway, so here, okay, same thing, that's a splicism. And ultimately, the axon will be placed together like this. Yeah, should use pink. That was our axon number 1. And that's the axon number 2. So they're placed together. Now, right now, this piece of, axon. So this piece here, we finally can call it a mature mRNA. So it can then move out of the nucleus through the nuclear pore for a translation process by the ribosome. So this bit here, the introns, will then, together with the splicism, will then be removed and destroyed. Bit ugly, but yeah, as long as you guys know what I'm drawing, I think it's fine. So yeah, it just fly away and bye. And so this will be gone. Okay, so will be degraded. So yeah, pretty much that's about it for splicing. So removing introns, placing exons together to form a mature mRNA, all right? So what exactly is alternative splicing then? Isn't it the same thing? So as I mentioned at the beginning of this video, alternative splicing is an application of splicing. What does that mean? So remember that I mentioned earlier that after removing the introns, we are trying to place the exons together. But in reality, there's not just 2 axons, right? There could be like lots of axons, with axon 1, axon 2, axon 3, axon 4. So is it really necessary to put all the axons together? The thing is, actually no. You don't always need to put all the axons together, because if you think about that, what exactly is the thing that's determining the amino acid sequence after all? It is the sequence on the mRNA. So if we can utilize different axons, so let's say for one mRNA, we use all the axons, and the next mRNA, we just selectively use some different axons, then we can ultimately kind of change up the mRNA coding sequence and make lots of different combo. I know it might sound a little bit confusing right now. So let me just draw a diagram to see if that makes sense. So, okay, another application here. So in reality, let's just say right now, this mRNA here, a little bit longer, okay? It has axon number 1. Let me just write E1, axon number 2, axon number 3, axon number 4, okay? And then some introns in between, like this, okay? So of course, after splicing, these introns will all be popped out, so they will all be removed. This is E1 again, E2, E3, E4. So using the understanding that we had earlier, so what we're expecting is that we are just going to place all the axons together. So yes, if we place all the axons together, and let me just scale it down a little bit because I don't have enough space, E1, E2, E3, E4, placing all of them together, E2, E3, E4. So this will be our mRNA coding sequence combo number 1, using all the axons, okay? So this would allow us, when the ribosome is reading the sequence on this E1, 2, 3, 4 combo, they're gonna be making protein A. Okay, because the sequence on the mRNA determines the amino acid that's coded, okay, that will be packed together to form that protein. So moving on, what is alternative splicing then? Alternative splicing is to selectively place different exons together to make new mRNA coding sequence combination, which means that in another situation here, we don't always, after removing the introns, okay, we don't always need to put all the exons together. Let's just say right now we're just using E1, E3, and E4. So we basically skip exon 2. And it'd be like, why do you want to skip exon 2? Isn't it like the meaningful part of the mRNA? The thing is, yes, it is meaningful. But if we are trying to make another protein, okay, what we have to do is just, if we alter the mRNA coding sequence a little bit, the amino acid coded will be changed as well, so we can make into another protein, which is a good thing, right? I mean, you can just use one pre-mRNA by cutting it and pasting it differently. We can be making different combo, making another protein. So here, call this exon skipping. Okay, so now we have another coding sequence here, so can make into protein B. So of course, in reality, there could be lots and lots of exons. So it's not just it's not just 4 exons here that's shown in the diagram. But yeah, which means that by selectively using different exons, placing them together, we're able to make different combo that really allows us to make and increase our protein size, all the proteins that we're making, expressing in the cell, okay, with a limited size of, we have fixed number of gene, around 20 to 25,000 gene. This is something that I always tell my students too. We only have 20 to 25,000 genes discovered by the Human Genome Project. However, we can actually make more than 1 million different types of proteins. How do we do that? And that's really because of alternative splicing. It's never just one gene okay, let me rephrase this. It's not just about using one gene. One gene is not just limited to making one protein. Okay, so you can use one gene to make multiple different proteins by just in this really carefully controlled process, alternative splicing, by cutting all the useless bit out, cutting all the introns out here. After cutting all these introns out, you can selectively place these E1, 2, 3, 4 in a different sequence. You can use all of them like this one, E1, 2, 3, 4, placing all together, making protein A. You can also skip E2 and make it into protein B, or you can, okay, another example here, or you can skip E3 and we can make it into what? E1, E2, E1, E2, and E4. So now we have another new mRNA coding sequence. New mRNA coding sequence. So that will be made into protein C. Fun time! We can make lots of protein with just one pre-mRNA molecule. Isn't it cool? It's so cool. So yeah, once again, it is done by Splicing. Okay, so yeah, that's really about it for alternative splicing. I hope you guys found some values in this video. If there is any question and you still don't quite get what exactly does it mean by alternative splicing, feel free to leave a comment down below. Yes, and I will see you guys next time. Bye-bye. Oh, if you're watching this in Hong Kong, stay safe. Okay, bye-bye.